Hello students, in this module let us talk about the wobble hypothesis. I told you there are not 61 anticodons of tRNA that is found in your cell, and in this module we will see how is that possible. Okay, look, listen and learn. So, what is the wobble hypothesis? The wobble hypothesis was suggested in the year 1966 by none other than Francis Crick. And according to this hypothesis, they say that in the codon-anticodon pairing, the third base may not be complementary. Let us see how. Okay, so I'm going to show you an example of a codon here, GUU. And if you read this from 5' to 3', you know that GUU codes for valine. You can look above me there. Okay, so if GUU codes for valine, then the tRNA will bring valine to the site. Now look at the anticodon of tRNA. It has to be CAA. Remember, because anticodon and codon is complementary. Let us see if the codons and anticodon bases are complementary. And we're looking at all three positions here. Look at the first position, G and C. Do you think it is complementary? Absolutely. G always pairs with C. So the first position is complementary. Look at the second position, U and A. Do you think they are complementary base pairs? Absolutely right. They are complementary base pairs. So here the second position also works fine. Look at the third position. Once again, U and A. You think they are complementary? Absolutely. So when all three bases are complementary to each other, of the codon and the anticodon, we call this kind of pairing as perfect pairing. Is that okay? Now I'm going to show you how that same tRNA, right, with the same combination of anticodon CAA will also bind or pair up with GUC, GUA and GUG. Let us see next. Okay, now look at this next combination GUC of the codon. You know that reading from 5' to 3' and looking at that chart above me, GUC also codes for valine. So that same tRNA is going to bring back the valine. See the same tRNA, look at the anticodon, it is still CAA. This time look at the three positions and the pairing between them. Do you think G and C pair with each other complementary? Absolutely they do. So we say that the position 1 is absolutely okay. Look at the second case, U and A. Is that complementary? Yes, it is. It is also complementary. But look at the third position, C and A. I doubt. I don't think it is complementary. So third position is not working right here. But as long as position 1 and 2 are complementary base pairs, we are okay. Let's see one more scenario. This time it is GUA. Once again, reading from 5' to 3', you know that GUA will also code for valine. Look above me. And now I'm going to bring in the same tRNA. Yes, with CAA anticodon. And now we look at the three positions and the complementary match between them. Position 1 is once again G and C, which is perfect complementary base pairing. Position 2 is U and A. Once again, a perfect complementary base pairing. Position 3 is A and A. You know that two A's will never complement each other, right? So the third position is not complementary. Okay, now looking at another situation, or the last one, GUG. Once again, reading from 5' to 3', you know that GUG also codes for valine. Now once you read GUG, the same tRNA brings in valine. Okay, once again, let us see the three positions and the complementary base pairing between them. You see that G and C perfectly pairs each other. So position 1, complementary base pair works. U and A also is base pairing with each other. So position 2 also complementary base pairing works. But the last position once again is doubtful because G and A do not base pair with each other. So there is no complementary base pairing at the third position. Is that okay? So in all these three cases, what you see is that third base up there is not matching or not complementary with its base on the anticodon. So this third base is called as the wobble base. And this pairing of the third position in all three cases is called as the wobble position. Is that okay? And what do they say here? That the actual base pairing occurs only at the first two positions. As long as they are complementary, the same tRNA can bind to all different type of codons. Is that okay? So, just summing it up, I'm showing you the same 4 RNA possible codons and the same tRNA binding to all 4 of them. And what do we see here? Though the codon, anticodon do not match perfectly, valine is brought perfectly by the same tRNA. Is that okay? You can see that here. And what would this help with? This will help with enabling the economy of tRNA, which means we can save on the tRNAs. We can have that same tRNA bind at 4 different positions, bringing the same amino acid every time. As in this case, you see that GUU, GUC, GUA, and GUG all 4 code for the same amino acid, which is valine. Okay, and this brings us to the end of this module where I talked about the Wobble hypothesis. In the next module, I'll begin with a very important process which is called as transcription. Till then, thank you and take care.